IB Biology class. This is Ms. Shealy with your next installment of your video notes. Today we are going to be doing 2.7 DNA replication, transcription, and translation. As always, make sure you take good and copious notes and write down any questions you have along the way so that we can review those when we get back to class. First thing we want to do is review why we need DNA replication. So we use DNA replication. It occurs during interphase of the cell cycle so that we can go through metaphase and telophase and separate the nuclei. So DNA replication is going to involve the sister chromatids, which are the chromosomes and their copies, and they are going to divide later through mitosis. So this is going to ensure that each daughter cell gets the correct number of chromosomes and that they are able to align and separate properly so that when the cell divides finally at cytokinesis, there's a full set of chromosomes at each pole forming the new nuclei. So how does it happen? Well, the first thing that has to happen is our DNA helix needs to unwind and unzip. So we use an enzyme called DNA helicase to do this. And you can see in this picture here, we have our double-stranded DNA twisted into the helix, and then helicase comes in and exposes the bases on each side. All right, so DNA replication. So the next step after helicase comes in is that the DNA polymerase is going to work on each parent strand to create the complementary strands. And you can see here that the complementary strands are being built in opposite directions. This one is going towards the helicase and this one is going away. The free-floating nucleotides are collected by the DNA polymerase and they're kind of just floating around in the nucleus so that they're available for base pairing. All right, so following our normal complementary base pairing rules, our adenine going with thymine and our cytosine going with guanine, the parent strand is going to act as a template for the new or complementary strands, and this is going to allow for DNA replication to be semi-conservative. This is going to ensure that our DNA molecules, the two daughter cells, daughter nuclei, are identical to the original. This is also going to help to ensure that no mistakes are being made because we are using this sequence and using our base pairing. Those nucleotides are going to be conserved. So if you'd like to go through, this is a little link to a DNA builder on Learn Genetics, and you can build and practice your complementary base pair binding. All right, so we talked about DNA being semi-conservative. Well, what does that mean? When our new strands contain one original and one copy or complementary strand, this is called semi-conservative. And how do we know that this is true? Well, we learned this through this is Matthew Messelson and this is Frank Stahl. And they were the ones that show that DNA is made by copying from the old. So we must make new DNA whenever a cell divides. Each daughter cell must receive a faithful copy of the parent cell's DNA. So Watson and Crick, in their final paper where they suggested or proposed the structure for DNA, they suggested that during replication the DNA strands separate and each strand acts as a template, but they weren't really sure how this happens. And Messelson and Stahl were able to show that it was this semi-conservative. So in 1958, Messelson and Stahl published results that supported this model, and you can see from their experiment and also an activity that we do in class that the clever use of nitrogen isotopes and a density gradient that was created through centrifugation. So they started first by growing E. coli in a culture with a nitrogen isotope, nitrogen-15, which is a heavy isotope. And as the bacteria grew and divided, the nitrogen-15 was incorporated into each of the nitrogen bases. It was incorporated into the A's and the C's and the T's and the G's. They saved a sample from this nitrogen-15 labeled bacteria for later analysis. And the rest of the culture was transferred to a fresh culture media containing normal nitrogen, which is nitrogen-14. This is a much lighter isotope than the nitrogen-15 isotope. So when the nitrogen-14 was available for subsequent construction of the DNA, the bacteria was replicated using this new nitrogen. And then we withdrew the cultures samples from the nitrogen-14 each time the bacterial population doubled. So this would be the first replication. And then the second replication. So for comparison purposes, they created a third culture of bacteria that was grown only in the nitrogen-14. So then they examined the nitrogen composition of the bacterial DNA from each culture, and we did this by isolating the DNA from the bacterial cultures, dissolving the DNA in a strong salt solution, and then they centrifuged the DNA at a very high speed. So the centrifugal force pulls more of the salt molecules towards the bottom, leaving fewer and fewer molecules towards the top. And the greater the number of the salt molecules at any point in the tube, the greater the density of the solution. I submitted attendance, but it wasn't right. Like, I just submitted everybody because they just called in. So, yep. Yeah, that's what I figured.
They put a physics textbook in my box with my name, like, specifically sticky noted on it. I don't know why.
That's weird.
So.
Yeah. Hey, Boo.
I don't know if this is a good time or not, but I was just asking you when it would be an awesome time for you to help me out with some bath stuff.
Yeah. Could we do today's Tuesday, Thursday during this time? Or is that too late for you?
I mean, I think you could do Thursday after. So should I just come in here and you'll
Yep.
be here? Okay.
That'd be perfect.
Awesome.
Cool. Thank you.
All right.
So we expected the DNA molecules that were made of the heavy nitrogen-15 would sink further in the salt gradient than compared to the DNA molecules of the nitrogen-14. And that's actually exactly what they saw when they looked at the samples drawn from the different cultures. So you can see here that the nitrogen-15 was much lower than the nitrogen-14. And this is, of course, the comparison. So these are the two controls, the start and the one that was only ever done in the nitrogen-14. But the two tubes in the middle were the two critical tubes, and they were containing the culture that was first grown in the nitrogen-15 and then switched to nitrogen-14. So this is sample 2 is after the bacterial population doubled once, and then the DNA band that they saw was exactly in between. So this showed that it had equal amounts of nitrogen-15 and nitrogen-14. And then sample 3 was taken after the second replication, and they saw 2 bands, and one lined up exactly with the nitrogen-14, and the other was halfway, just like in tube 2. So this DNA perfectly matches the results predicted by the semi-conservative DNA replication model. The heaviest DNA molecules were found in sample 1, where both of the strands were made in the only nitrogen-15 isotope. The lightest DNA molecules were found only in sample 4, and remember that DNA was only nitrogen-14 because the bacteria were only grown in the regular nitrogen-14 isotope. In sample 2, the DNA molecules collected as a band of intermediate density. So when the bacterial culture was switched from the 15 to the 14, any new DNA made was only using the nitrogen-14. So the two heavy strands of the parent DNA separate, and they act as templates for the incorporation of nitrogen-14 into the new strands. So all the DNA are hybrids with one heavy nitrogen-15 and one light nitrogen-14. And then sample 3 is where the DNA went through a second round of replication. So we have 2 separate bands, and in this case, each hybrid molecule separates into one heavy and one light strand, and the heavy parent acts as a template for making the complementary light strand, resulting in another hybrid. And the light parent strand is also a template for incorporating light nucleotides. But this time, the resulting molecule is 2 light strands. So you're going to have that intermediate density DNA and the light density DNA showing up as 2 separate bands. So they didn't know the details of how this DNA process replicates when they published their results, but it was very clear that Watson and Crick's predictions about the semi-conservative replication of DNA were correct, and their experiment did falsify the idea of the conservative DNA model and the dispersive replication model. So there's other times where we need replication to occur, and we actually have a clinical application of replication, and that's called the polymerase chain reaction. PCR can be utilized to make many copies of DNA. We use this in both gene transfer and when samples of forensic science are very small, have very small amounts of DNA. There are basically 4 steps in PCR, or polymerase chain reaction. The first step is that we heat the DNA, and this is so that it artificially splits open. We cool the DNA so that the primers can be added. The DNA is heated again for a special DNA polymerase to come in and build the complementary DNA strands. And that type of polymerase is called Taq polymerase, and it's from an organism called Thermus aquaticus, which lives in hot springs and therefore does not denature in high temperatures. And this process is repeated over and over and over until we have millions of copies of DNA.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
All right, moving on from replication, we're going to move on to making of proteins, which involves transcription and translation. So first we need to understand or review the central dogma of genetics, and that's that we go from DNA, where our genes are, to leaving the nucleus, to making the RNA, which is our message. That's a process called transcription. We go from the message of RNA into the cytoplasm and make our polypeptide product, which is a process called translation. So remember that genes are just simply codes for making polypeptides. The DNA is stored in the nucleus, yet the polypeptide is produced in the cytoplasm by the ribosomes. So our mRNA messenger RNA is a message from the nucleus to the ribosome and instructions for how to put this polypeptide together. The genetic code is then a sequence of bases on the mRNA, and this tells the ribosome which amino acids to use to put together our protein. So the first thing that we need to make our proteins is a process called transcription, as this is when we use an enzyme named RNA polymerase. He works very, very similarly to DNA polymerase, and he makes the mRNA molecule that's complementary to the DNA. And then, once we have the mRNA sequence, we'll use a process called translation, which is when the mRNA is posted out of the nucleus and attaches to the ribosomes. The ribosomes use complementary base pairing to read the codons on the mRNA, and then the tRNA transfer molecules with the corresponding anticodons bring the correct amino acid in. So here, let's look at transcription first. So the first thing we have is our RNA polymerase comes up to our DNA. It spots a region called a promoter region. And at that promoter region, it binds and opens. As it's opening, it's able to build the mRNA molecule. There's no other enzymes that are needed. RNA polymerase is able to do it all. It continues until it gets to an area called the terminator, and that area tells the RNA polymerase to stop. Now let's look at translation. In translation, we have a small ribosomal subunit that docks the mRNA to it. The tRNAs come in, and that signifies that the large ribosomal subunit can come in. Notice that there are 3 different spots on the large ribosomal subunit. These are areas. They're not actually having labels, but there are areas that we as scientists have labeled as the E, the P, and the A site. The first anticodon will dock onto our start codon, which is AUG, and this is a tRNA that's containing the amino acid methionine. He's going to dock onto the P site, leaving the A site open for the next amino acid and tRNA to come in. The amino acid's going to leave the first tRNA, bond to the second tRNA, forming a peptide bond between. That's going to allow the ribosome to scoot down or move down the mRNA so that the 2 tRNAs are now in the E and the P site, leaving the A site open. The tRNA that's in the E site will then leave, and the A site is now open. So we'll continue this process where the next one comes in to the A site. The amino acids bond to this amino acid attached to this tRNA, and it continues repeatedly. Till we get to an end where we get a protein called a release factor that comes and allows the ribosome subunits to break off from the mRNA. All right, so how does the ribosome know which amino acid to add next? So as we just saw in the sequence, the ribosome moves down the mRNA. It reads the codons on the ribosome, and then the tRNAs come in. So the anticodon on the tRNA is the complementary sequence to the mRNA codon. So the tRNA molecules have very specific amino acids. They can only each carry one type of amino acid. All right, so a little challenge for you. Each of these string of letters has a sentence in it. Can you find the sentence in the string of letters? So here's the sentence highlighted for you. I hope you each found them. So the first one was the man was too old for the war. The second one, the boy had one red car and one old hat. A little difficult to read. And the last one is the car was red and too old for her new job. So, but what do they have in common? Well, if you notice, they're all 3-letter words, and this example is very similar to how DNA is transcribed in triplets, which is a sequence of 3 bases, and this gives the mRNA codons, which is sequences of 3 RNA bases. And those mRNA codons make up the genetic code. Each sentence also starts with the same word. In this example, each sentence starts with the, and that is because each gene starts with the same start codon, and that is AUG, so we all start with the same amino acid, which is a methionine. All sentences also finish with end at the end of the sentence, and it's not actually part of the sentence. If you were to read the first sentence, the man was too old for the war, end, doesn't really make sense. The end signifies where the punctuation would go, where the end of the sentence goes, and in this analogy, it would represent the stop codon at the end of the gene. There are many words and phrases that are repeated or reused, and this is equal to or represented by the 64 different combinations, but allow for infinite possibilities of polypeptides. So these many words are reused and reused and reused. The really big difference is that our sentences used all the letters of the alphabet, or could use all the letters of the alphabet, and our genetic code only uses 4 letters. It only uses A's and T's and G's and C's. So this shows that our genetic code is universal. All living things use the exact same code. They all use the A's and the T's and the G's and the C's. And they, moving to the mRNA and tRNA, utilize the U's instead of the T's. They use the same amino acids and anticodons and codons combinations. And it does not matter which organism it is, whether you're a banana, a monkey, a pea, a human, everyone uses the same code. There are 64 codons, but there's only 20 amino acids plus stop codons, or plus codons that represent stop, which is not an amino acid. So the same amino acid may be coded for by more than one codon. If you look in this chart over here, we have actually 6 different codon possibilities for leucine. So this reduces the impact of base substitution mutations, as the amino acid produced could still possibly be the same no matter what the codon combination is. Other things to remember about the DNA is that in this particular example, our gene would give us 21 base pairs, but this would only give us 7 codons. And when we're making our polypeptide, this would only give us 6 amino acids. Another thing to remember, as previously stated, that methionine is always the first amino acid, so your codon that is always first is always AUG. The number of amino acids to codons is 1, and the number of amino acids to base pairs is, there's 3 base pairs to every amino acid. So remember there are 20 amino acids and stop codons. And that's it for today. Hope you took notes and bring your questions to class.